and a former postdoctoral researcher at the I3S, Institute for Research and Innovation in Health, University of Porto, Portugal. Daniel has been working on different aspects of redox biology and oxidative stress throughout his academic career. In his current position, Daniel works on the discovery and characterization of bioactive peptides from amphibian skin secretions, with a particular emphasis on peptides with antioxidant properties. His work aims to identify candidate peptides for further development into therapeutic agents for oxidative stress-related conditions. I would like to invite Dr. Daniel Moreira to start his talk. Good luck.
Okay. The pointer. Can we change it to the laser thing? Oh, thank you. já, já está, já está. Okay, thank you. Thank you, Professor Mariana, for the invitation, the organizing committee, and you all for being here so far. I'm Daniel. I work as a biologist in the Research Center for Morphology and Applied Immunology under the supervision of Professor José Roberto Leite. And today I'll talk about some antioxidant peptides from amphibians that were collected in Brazil and Portugal. And that's it. They have antioxidant activity, and we are interested in that. And before talking about the peptides, I'd like just to present a brief background on redox biology. And I always like to start with the oxygen paradox, which is the title of this book published in the 90s that explored this idea that aerobic organisms are highly dependent on oxygen, yet oxygen can be highly deleterious. And the reason is that, as all of you are aware, is that oxygen can produce reactive species like free radicals. And actually we have multiple enzyme systems that do so in animal cells. We have several systems that are able to produce reactive species, and also other systems that are specialized in produce specific reactive species. And this happens at multiple sites inside a cell, not only in the mitochondria from the electron transport chain, but also from others in other sites inside a cell. And luckily, aerobic organisms have developed or evolved over time antioxidant systems to manage these reactive species. So on the left, we have a big picture of what is redox metabolism, of redox biology. We have sources of reactive species, such as NADPH oxidases and the mitochondria, other oxidases, and the exposome that here includes exogenous factors such as radiation exposure, nutrition, and other exogenous factors. We have redox-sensitive proximal targets that, for example, these are very well-known transcription factors that are sensitive to redox changes. They trigger downstream signaling events. We have also some proteins that control the diffusion of hydrogen peroxide, for example. And we have the antioxidants that act as sinks. They neutralize and consume reactive species. And here on the right, we have the classical core endogenous antioxidants and the most important reactive oxygen species. We have here a superoxide radical, which is dismutated by superoxide dismutase, generating hydrogen peroxide. Hydrogen peroxide can, in the presence of transition metals such as iron and copper, can generate the highly reactive hydroxyl radical, or it can be decomposed by catalase or other antioxidant systems, such as glutathione-dependent systems, like depicted here in this scheme. We have glutathione peroxidase that decomposes hydrogen peroxide, and in this process, it oxidizes glutathione. And on the upper part here, we have the thioredoxin-dependent system that also decomposes hydrogen peroxide, but oxidizes thioredoxin instead of glutathione. And both the systems have its own reductase that needs NADPH to keep this thing working. So this is the core antioxidant systems, the main endogenous antioxidants in an animal cell. And under physiological conditions, what we have is that we are continuously producing reactive species under the control of antioxidant systems. And through the modifications in iron sulfur centers and cysteine modifications, cells transmit signals in what some authors call the redox code. And this is completely normal to have oxidation of cysteines. This transmits signals to the cell, and ROS and other reactive species are absolutely necessary for aerobic life. And it participates in key processes from immunity, development, cognition. But there are some conditions in which this relationship between production and neutralization of reactive species is disrupted. So we have an excess of reactive species that can oxidize DNA, proteins, and other biomolecules, usually causing loss of function. And this loss of function is associated with many pathologies. And if we look at the list of diseases that have a redox imbalance component associated with it, it's a long list, but here we have just an example. For example, the ratio between the oxidized form of glutathione to its reduced form, alterations in this ratio, this is a very good marker of redox balance. It happens in all of these diseases, from cardiovascular disease, neurodegenerative conditions, cancer, and the list goes on. But we also have other markers of oxidative stress, such as oxidative damage to proteins, such as protein carbonyls and nitrotyrosine. We have damage to lipids, such as hydroxynonenal, MDA, and isoprostane, and oxidative damage to DNA. And the list is so big, and it's always increasing, that the authors of the main textbook in redox biology, they have just gave up to compile this list. There are just too many disorders or pathological conditions in which reactive species have been implicated. And they argue that changes in redox balance, reactive species, and antioxidants are related to all human diseases. And this is from where the interest in antioxidant molecules came from. So there are many research groups around the world looking for antioxidant molecules to treat diseases that are associated with oxidative stress. And one of the sources of antioxidant molecules is the skin secretion of amphibians. They have not only the well-studied antimicrobial activities, but they also have antioxidant peptides. And a Chinese group in 2009, they have actually proposed that the antioxidant peptides in the skin of amphibians also actually are a whole new antioxidant system that would act in concert with the core endogenous antioxidant system I showed before. And the reason for that is that the skin is highly vulnerable to infections, which is associated, again, with oxidative stress, to exposure to direct exposure to air containing oxygen, and also exposure to ultraviolet radiation. So these authors it's the first time that the antioxidant peptides in the skin secretion of amphibians are treated as this new skin antioxidant system. So, in our lab, our typical approach to study antioxidant peptides is first with the identification. We have two approaches. We have the HPLC fractionation of crude secretions, followed by mass spec sequencing, or we work with the cDNA analysis, and often we combine both. So after peptide identification, we proceed to synthesize it in the lab. We purify the product of the synthesis, and we start with the in vitro chemical assays. Basically what these assays tell us is the ability of the peptides to engage in single electron or hydrogen atom transfer reactions. Then we proceed to still in vitro, but using cells. We usually test cytotoxicity with red blood cells and also in other cell lines. And we have several systems to induce oxidative stress to the cell and see how the peptide affects this process. And in some of the peptides, we proceed to in vivo experiments, but we don't have many results from these in vivo studies. So our work with antioxidant peptides started with antioxidant, which was a peptide found in the skin secretions of Physalemmus nattereri. Here's the chromatogram. And after sequencing, we first did the obvious, that was to test its antibiotic activity, and it was not very exciting. And we tested these strains. And one interesting thing we found was that from our cDNA bank, we found that this peptide was also found in the skin cDNA of other 3 tropical frogs. Then we decided to test, well, if the peptide is not antibiotic, let's test its antioxidant activity. And what we did was to perform the chemical-based assays, and we used 4 assays. And basically what we see, what these experiments do is for ABTS and DPPH, these are artificial free radicals that have a strong color. You can see here in this picture, ABTS is green, DPPH is purple. And if you have a molecule that neutralizes it, it loses its color. The ORAC assay is an assay based in the ability of the molecule to protect a fluorescent protein, and we generate free radicals chemically and check if the peptide can inhibit the oxidation of another molecule. And we also measure the ability of the peptides to scavenge nitric oxide. And in this first study, we also synthesized the antioxidant RP1, which is a peptide that was reported in the literature as the most antioxidant of peptides amongst the peptides from amphibian secretion. And when we compare the performance of our peptide with the antioxidant RP1, we see that our peptide did not do very well. The peptide from the Chinese frog, its RP stands for Rana pleurodane. And we see that the peptide from the Chinese frog has a much better performance than antioxidant, with the exception of the Oracacea. So this motivated us to further explore it in cell-based experiments. We first checked if our peptide was not cytotoxic to the cell line we would use. And then we tested the ability of the peptide to prevent oxidative stress caused by menadion. And what menadion does is it causes a redox cycling reaction inside the cell, and the cell constantly produces superoxide radicals. As we can see here, we could successfully induce a redox imbalance. We have increased the ratio of the oxidized to the total glutathione content of the fibroblast. And in the presence of the peptide, this was prevented. So showing that at a fairly high concentration, but still it prevented the oxidative stress caused by menadion, sorry. And we also did some controls because menadion is soluble in ethanol, but the overall result was that antioxidant 1 prevented the oxidative stress caused by menadion. Then we advanced to another cell type. We used HMC3 human microglia, and we transfected these cells with hyperRAD, which is a biosensor for hydrogen peroxide. And here we, instead of using menadion, we put cells in hypoxia, low oxygen levels. And what we see here in this upper line is that over time, as the time goes by under hypoxia, cells produce more hydrogen peroxide. And in the presence of antioxidant 1, this pretty much nothing happens. And this is a representative result for one cell, but when we take together the fluorescence intensity for many other cells, we have this graph showing that antioxidant 1 prevented the increase in ROS production in response to hypoxia in microglia. And that's where we are at with antioxidant 1. And now moving to the next peptide. We also collected some animals in Portugal. This was one of them. We collected the specimens from the fire salamander, the species salamandra. And we could sequence this peptide here in the chromatogram. It's a 12 amino acid peptide. We also performed the BTS and DPPH assay. And here we compared with glutathione, which is an extraordinary antioxidant endogenous, just for comparative purpose. But these results of 0.285 is pretty good if we compare to the other peptides. Oh my God, what did I do? Okay, it's back. Here, okay, so we have 0.3 for antioxidant RP1 and 0.285 for salamandrine 1. And one interesting, we didn't advance much with this peptide, but it's interesting that this was when we published this paper, it was the first peptide that had been identified in the skin secretion of the Salamandra genus. And we proceeded to the cytotoxicity test. The peptide does not cause any hemolysis in human blood cells, and it's not toxic to HMC3 human microglia. And we also tested, which was well accepted at the time. We used it for the first time, at least in our studies, the insect model for in vivo toxicity. So we simply injected the peptide in the animal and checked their survival. We didn't stress them with any other thing. We just wanted to see if the peptide was toxic. And we did some histological analysis showing that animals did not die in response to salamandrine 1, and we did not find histological alterations in key tissues such as the fat body, the gut, and the cuticle. And that is where we are at now with salamandrine 1. Now for the next peptide. So this is a Portuguese specimen too. We collected Pelophylax perezi in Ilha dos Açores, and we could sequence this peptide. It was named PPT2 because it was classified as a tryptophan because of this PW motif. And what we saw again was a fairly good ABTS performance, a 0.269, and a very low performance with the DPPH assay. And one interesting thing is that when we also checked our cDNA bank, we could also find this sequence in the cDNA of Physalemmus centralis, which was a specimen collected here in Tocantins. And pretty much is following our usual approach. We test for any hemolytic activity. We didn't find anything significant. We also test the viability with HMC3 human microglia with no cytotoxicity. And we also did the in vivo toxicity using Galera melanella larvae, and the peptide was safe. And what we did, we used BV2 mouse microglia, and we induced ROS production with PMA. And what PMA does, it's a protein kinase C agonist, and it activates NADPH oxidase, so the cell produces more reactive species. And this is what is shown here. We have the control in gray and the PMA-treated cells. We measure reactive species using the fluorescent probe, DCFDA, and in the presence of the peptide at 50 and 100 micromolar, this effect was abolished. And when we also measure reactive nitrogen species using the FFM, we also had a similar effect. We effectively increased the production of reactive nitrogen species, and the peptide was able to reduce this increase. And at the highest concentration, even the basal reactive nitrogen species production was reduced. And that's it for PPT2. And for the last one, and PAT2 was a peptide. It's also a tryptophan. It was classified as a tryptophan because of this PW motif here. It was found in the secretion, as Professor Guilherme explained yesterday. When we have these prolines, we have the same peptide in multiple peaks. And this was the skin secretions of Pithecoppus azureus. And one interesting thing about this peptide is that we tried to find it during the ontogenetic development of this animal, and we tested animals at different time points during metamorphosis, and using both MALDI imaging and analyzing the skin secretions using HPLC, we found that this peptide begins to be expressed when animals are at this stage 41. And this is a very critical stage for the animal because it's the stage that precedes the terrestrial behavior of this species. And during the molding imaging analysis, we looked for peptides, and the only one we could find was PAT2. And this was confirmed in the skin secretion, and we found that this species secretes a single peptide right before going to land, when it leaves water to the land. And this was enough for us to believe that it would have antioxidant properties. Because when animals leave the water, they are more exposed to external factors such as UV radiation, and the oxygen concentration in air is much higher than in water. And then we again tested this peptide in BV2 mouse microglia. And here we use again PMA. We successfully increased the production of reactive oxygen species, and the peptide PAT2 at 50 micromolar or 100 micromolar could inhibit this overproduction of reactive oxygen species. And here again, at the highest concentration, it could reduce the basal production of reactive oxygen species. And once again, we also measure the production of reactive nitrogen species using the same cell line. And we have pretty similar results. The peptide can prevent the increase in production of reactive species at 50 micromolar and 100 micromolar. And here for the last slide, this is wrong. This is for PAT2, not an antioxidant, sorry. And what we did was the same experiment with the gene-encoded hyperred probe for hydrogen peroxide, but this time we used LPS that triggers our inflammatory response by microglia, which includes both the production of reactive species and the release of glutamate, which is excitotoxic. So we saw that the peptide could decrease the production of reactive oxygen species in response to LPS, and it was a significant difference, but very small one, reduce the release of glutamate when the microglia was treated with LPS. So to wrap it up, we are far away from developing a product yet, but we can say that we have a rich source of antioxidants in skin secretions. Their structures, their primary structures are very different, but we have low cytotoxicity, and we have shown the ability to reduce levels of reactive oxygen species and nitrogen species in cells, not in vivo. And we believe that these peptides, even if not these exact molecules, but as a template for the development of other molecules, they might be useful. And of course, we have not even started to understand the mechanism of action. You only know that they decrease ROS, but we have no idea of at the molecular level how this happens. And of course, we have very little in vivo studies with these molecules. And I'd like to thank our partners inside University of Brasília, our partners in São Paulo, in Portugal, and our funding agents. And thank you for being here so far.
Congratulations, Daniel, for a very well done work. Your data are very interesting, and I would like to open for questions. I have one. Sorry.
Are you interested in test with pathogenic fungi? Are you interested in testing with pathogenic fungi?
Oh, no, we have not tested these peptides against any. Only for antioxidant 1, we tested with some bacteria, but not fungi, no. But we have the molecules. About the effect of these peptides in wound healing. We don't have any data. We have there's a strong Chinese group that is working with antioxidant peptides, and they have some good results for wound healing and for protection against UV radiation. And they attribute this effect to the antioxidant properties of the peptides they have tested. But we have not done any experiment in that matter.
And now I would like to close the activities for today. And tomorrow we will start at 8:30 with the symposium coordinated by Professor Fernanda Cardoso. Okay, see you tomorrow. Have a good night.